Assalamualaikum warahmatullahi wabarakatuh. So here is the video of our group presentation, which is the production of penicillin. So what is antibiotics? Antibiotics are powerful medicines that fight certain infections and can save lives when used properly. They either stop bacteria from reproducing or destroy them. There are several antibiotic resistance, which is bacteria changing in ways that reduce or eliminate the effectiveness of antibiotics due to bacterial evolution. So what is penicillins? Penicillins are antibiotics or a group of antibacterial drugs that attack a wide range of bacteria. They were the first drugs of this type that doctors used. The discovery and manufacture of penicillins have changed the face of medicine, as these drugs have saved millions of lives. The function of the penicillin is it bursts the bacterial cell walls. They do this by acting directly on peptidoglycans, which play an essential structural roles in bacterial cells. There are several resistances of penicillin. Bacteria are sometimes able to produce penicillinase, an enzyme that degrades penicillins. And some bacteria can subtly change the format of the penicillin binding proteins in their peptidoglycan walls so that penicillins can no longer bind to it. Something interesting to know about the penicillin. Do you guys know that the penicillin was the first antibiotic? Let's know more details on how the penicillin was made. The first name for penicillin was moldjuice. A Scottish bacteriologist, Alexander Fleming, accidentally discovered the antibiotic in 1928. He came back from a vacation and found that a green mold called Penicillium notatum had contaminated petri dishes in his lab. The green mold were killing some of the bacteria he'd been growing. He isolated the mold, grew more of it, and then experimented to see how many other bacteria it could kill. He now knew that penicillin works by preventing bacteria from forming new cell walls. There are three workflows of how penicillin was made. The first one is upstream, the second one is fermentation, and the last one is downstream.
The upstream process of penicillin. Upstream process referring to process before input to the fermenter and encompasses any technology that leads to the synthesis of a product. It includes exploration, development, and production. Production method. First, the secondary metabolites are only produced in times of stress when resources are low and the organism must produce these compounds to kill off its competitors to allow it to survive. Second, it is these conditions that we wish to duplicate in order to achieve the maximum amount of product from our fermentation. This can be seen in the graph above of primary metabolites and secondary metabolites. In the stages of production, primary metabolites will be emphasized. Media for this stage will be focused on achieving maximum growth and biomass production. Once the desired biomass has been achieved, stop limiting the amount of carbon and nitrogen available to the culture and induce the kind of stress conditions that trigger the production of the antibiotic. The production of penicillin was the first important commercial product produced by an aerobic submerged fermentation, first antibiotic to have manufactured in bulk, and at the end of World War II, penicillin was first made using fungus, Penicillium notatum, which produced a yield of 1 milligram over m squared.
A fermentation process starts in a fermenter. A fermenter is a type of bioreactor for containing and controlling fermented microorganisms. The purpose of a fermenter is to provide a contained, controlled, homogeneous environment in which the fermentation can proceed in a manner that is both safe and practical. Typically, all microorganisms require hydrogen, carbon, sulfur, oxygen, and nitrogen for cell growth and cell maintenance. When performing any kind of fermentation, the selection of media or nutrient is of critical importance to the overall performance of the fermentation. The aim of the media or nutrient is to provide all the elements required for the synthesis of cell materials and the formation of the desired product. The fermenter controls the pH, temperature, and the pressure of the bioreactor. The pressure is controlled by the cooling jacket to prevent overheating. The mortar is used to stir the culture, and sterile A is to prevent contamination by other microorganisms, and pressure release valve is to prevent the expulsion caused by excess gas during fermentation process. All of these are monitored to provide the optimum growth of the culture and to prevent explosion. The products are drained via the tap before the downstream process.
Now let's move on to the downstream process. Downstream process is a multi-step procedure for recovery and purification product of interest. This process occurs after fermentation process. In downstream process, there are several steps involved in production of penicillin. These include filtration, addition of solvent, centrifugal extraction, fluid bed drying, and storage. Firstly, filtration process is carried out to remove the biomass from the culture. In this process, rotary vacuum filter is commonly being used for filtration as it is able to run continuously in any large-scale operation. After filtration, phosphoric acid is introduced to decrease pH from 8 to 6.5 to prevent the loss of penicillin activity. Next is the addition of solvent. The organic solvents such as amyl acetate and butyl acetate are added to dissolve the penicillin present in the filtrate. At this point, penicillin that present in the form of solution and any other solid will be considered as a waste. This penicillin can be used as fertilizer or for animal feed. Then, it is continued with centrifugal extraction. The liquid component containing penicillin is centrifuged to separate the solid waste. Usually, this centrifuge is used at this point. The supernatant is collected and mixed with sodium bicarbonate solution to produce a penicillium sodium salt, which allows penicillin to be stored in a stable powder form. The powder penicillin salt is then undergoes the drying process using fluid bed drying. The drying process are done to remove any remaining moisture present in the powder penicillin salt. The hot gas in the fluid bed drying is pumped from the base chamber, which results in a much drier form of penicillin. Next is storage. The penicillin is stored in a container and kept in a dry environment. The resulting penicillin can be chemically and enzymatically modified to make a variety of penicillin such as penicillin V and ampicillin.
So in conclusion, penicillin was the first important industrial product of anaerobic submerged fermentation, which are upstream fermentation and downstream process. So for the upstream process is referring to the process before input to the fermenter and encompasses any technology that lead to the synthesis of product. While for the downstream processing is referring to the process done to purify the output of the fermenter until it reaches the desired product, such as extraction and purification of product from fermentation. So penicillin is the first antibiotic to have been manufactured in bulk. Penicillin also was made by using Penicillium chrysogenum. So here is the structure production of penicillin. So that's all from us. Thank you.
Here are the list of reference for this video.